Hello, everybody. I'm Bruce McKern, an adjunct professor at the UTS Business School, and I welcome you to this very interesting session we're going to have with Professor Lucy Shapiro, calling from Stanford University. As you know, this is part of a program that's been developed in conjunction with UTS and the Stanford Alumni Association in Australia. And we've had a number of very interesting speakers. But this is the first occasion on which we've had the opportunity to hear from a professor who is one of the outstanding leaders in the world in the field of essentially molecular biology and the understanding of the development of the cell. So I want to welcome you all to this session. Professor Shapiro will talk to you in a few minutes. First, I'd like to acknowledge the Gadigal people of the Eora Nation, upon whose land of the UTS City campus now stands. We'd like to pay respect to the elders, both past and present, and acknowledging them as the traditional custodians of knowledge of this land. As I mentioned, this webinar is part of a unique partnership between UTS and the Stanford Alumni Foundation of Australia. And part of the objective of that foundation is to help people participate in not-for-profit activities at Stanford University. I welcome, for that reason, specifically members of the Stanford Australia Foundation who are guests in this session today, and also those people who are alumni of UTS Business School, UTS Faculty of Science, both here in Australia and around the world. Welcome, and we look forward to hearing from you today. I should mention that this is being recorded today, and it'll be available for you to see again shortly after the program concludes. I'd now like to introduce Professor Shapiro as our special guest today. We want to hear from her, so I'm not going to say a lot about her background, but simply to say that she is a remarkably successful professor in the field of developmental biology, in the field of molecular biology. And as you know, she is the Ludwig Professor of Cancer Research and Director of the Beckman Center for Molecular and Genetic Medicine at Stanford. She also was awarded the National Medal of Science for 2011 by by President Obama. And quite recently, she got the Dixon Prize in Science from Carnegie Mellon University. You can read more about her background and you'll find that it's extraordinarily interesting because not only is Professor Shapiro a scientist, but she's also a person who's developed two business startups, one of which was very successfully sold to Pfizer a couple of years ago. So without further ado, I'd like to turn the session over to Professor Lucy Shapiro. Lucy.
Oh, it's a pleasure being here with everybody today. The magic of Zoom, which is what we've been living under for a year and a half now. Why are we living under this bizarre kind of world that we live in? And that is because of the 2020 pandemic. We have now lived through 179 million cases of COVID infections globally, which has killed almost 4 million people. And what I wanted to do this evening, well, it's this evening for me, it's during the day for you, is ask and answer a series of questions. One is, why has this happened? Two, what do we know about the COVID virus and what makes it a pandemic strain? Three, what is the status of the COVID vaccines and variants that are popping up with alarming frequency? What is the origin of the SARS-CoV-2 virus, which is a very important topic right now? And then actually, most importantly, how do we prepare for future pandemics? Now, in the immediate years before the COVID pandemic struck, there was a parade of diseases that started popping up around the world. I'm talking about 5, 10, 15 years, all squinched together in one narrow time slot. such as West Nile virus, SARS-1, the first coronavirus, dengue fever, Zika virus, Ebola virus. Now, this uptick in new pathogens and old pathogens in new places was a clear warning sign that we had entered into a new era of infectious disease threats. We knew that a pandemic was coming. Nobody could quite conceive of the extent of the pandemic that it was coming, but it's here. So why are we faced with this increasing global infectious disease threat? What are the factors that contribute to this sudden appearance and redistribution of these pathogens? Well, obviously, if something pops up somewhere, we live in a global village. We have jet travel, global trade, and loss of borders that really do lead to situations where somebody gets sick in the one continent and they're immediately sick in another continent. We are living through a time of geopolitical upheavals and displaced populations so that you can rapidly send a pandemic strain around the globe because of our living conditions. Then the things that are really stimulating this are overpopulation, deforestation, and urban crowding. One of the most important and impactful things is climate change. So I think we all have to appreciate the fact that our planet is a biological ecosystem that is highly tuned to environmental fluctuations. And right now, the equilibrium of this system has been challenged. The Earth is not static. It's constantly changing over millennia. There have been cataclysmic events through millennia that have initiated changes that play out over very long periods of time. So ice ages and global warming usually play out along 10,000 years. And what usually then happens in climate change is that you wind up with mass extinctions. We are, in fact, in the midst of the sixth great mass extinction. Now, what is different now, and very different now, is that the rate of change of the world's ecosystem is very rapid in geophysical time. And it is we, humans, that have produced this cataclysmic event. So, how does climate change affect infectious diseases? All right. Climate change means you have warmer places, colder places, dry places, wet places. The entire weather pattern is disturbed across the entire globe. What happened is that changes in climate result in changes in the habitat of viral, bacterial, and fungal pathogens, and the vectors that carry them to the human population, such as rats, mice, bats, and importantly, mosquitoes. Now, what happens is that as you go up a very little bit in temperature and you have lots of wet places or lots of dry places, these vectors are moving. So that diseases that were once in a very tight tropical band are now moving up into temperate zones and even higher. For example, malaria is now getting up into the high Himalayas because of temperature changes. And so it was inevitable. We knew it was inevitable that a pandemic strain would arrive, and we knew it was coming. What we all thought was coming was a pandemic flu, influenza. We did not predict coronavirus flu, even though we had SARS-1 in 2002. So what in fact makes SARS-CoV-2 a pandemic strain? Several things. Number one, human-to-human transmission, most important. Droplets, coughing or sneezing, aerosols, talking, singing, exertion. Importantly, contagion, 4 days before symptoms and several days after symptoms abate. So you are infected before you're showing any signs of being ill, and you are highly infectious before you even know you're infectious. So this asymptomatic infection is one of the key parameters in this wildfire of spreading this virus around the globe. On top of that, we have super spreader events that will get hundreds, if not thousands of people emanating from a very small number to begin with. So now this all looks pretty grim, but let's counterbalance this damage to our planet that we're all dealing with. And that is that we are in the midst of a technological revolution that has changed what we are able to do in the biological sciences. What do I mean by that? Well, we now have access to the information stored in the DNA of all living organisms. This is a monumental technological breakthrough. We can read the instructions, and the instructions come in DNA in our genomes, which is then translated into mRNA, a copy, and that mRNA makes protein. And I'm going to come back to this. So I'm being a teacher. DNA is copied into mRNA, and that's the messenger RNA, which is then translated into the proteins, like parts of the viral surface proteins in COVID-2. Now, access to information will allow you to use this in a way that is critical to our survival. For designing therapeutics and vaccines, and I'll get to that in a minute. For the global monitoring of COVID-19 infections, which is critical. For the surveillance of emerging infectious viruses and other pathogens of humans, livestock, agriculture. So, it's not just us, it's what we depend on, which are our plants and animals on this globe. So turning to vaccines. All right, there are fundamentally 3 or 4 types of vaccines out there. One of them is based on that messenger RNA that I told you about. Go from DNA to mRNA to a protein that when it gets made elicits an antibody response, which then protects you. So when I listen to this and I know what has happened with Pfizer and with Moderna, I think it's so important that people realize that this didn't happen like Venus out of a clamshell. It didn't just pop into being. It's a new technology, but it's been being worked on for tens of years. This is not brand new. People have been trying to make vaccines from this messenger RNA for quite a while now. There was one gigantic breakthrough in that we learned how to stabilize the RNA and how to make it so that it wouldn't cause inflammation in the people it was injected into. So we now have a completely safe and highly effective vaccine that is produced both by Pfizer and Moderna. A second new vaccine is based not on the mRNA, but on the DNA that encodes a specific protein, DNA to mRNA to a protein. And the protein that you want to encode, both in the case of the mRNA and in the adenovirus-based vaccines, which is AstraZeneca, Oxford, Johnson & Johnson, and Sputnik 5. In those cases, what they've done is they've taken a little piece of the DNA instruction that ultimately codes for a piece of the spike protein that sits on the surface of the COVID virus. This piece of DNA is spliced into the bigger piece of DNA sitting in a safe virus that cannot replicate inside of us or make us sick. That's adenovirus. And as I said, AstraZeneca, Johnson & Johnson, Sputnik 5 are all adenovirus-based vaccines. Then we have vaccines that are in fact based on inactivated viruses, and those basically come out of China, Sinopharm and Sinovac. And just recently, there's been another kind of vaccine out of Cuba, where they have made purified protein from the virus that once it's injected into you, elicits an antibody response. Now, these are our vaccines. And often people say to me, you know, if I'm fully vaccinated, why do I have to wear a mask? I'm always asked this question. So let me get it over with right up front. When you're vaccinated against something, what you're doing is you're teaching your body to recognize that something, let's say the surface of the COVID virus, and make antibodies. So you make antibodies and you can help protect yourself once you're infected. It does not stop the infection. It just mobilizes a little army inside your body to protect you from getting very sick or sick at all. But you can still be infected and asymmetric and transfer those viruses, that low level of virus, but some virus, onto other people who are not vaccinated. So wearing a mask is really preventing the transmission of a possible infection that you may have to those around you. So it's a good idea to have that. So now, one of the really important points and big breakthroughs about having these new vaccines based on RNA and DNA, however they get into your body, is that now that you know how to make it, if you get another virus or a variant that has a somewhat different sequence, it takes 2 days to design a new vaccine that will then be protective for you. The holdup is not in designing the vaccine and even making it, it's scaling up and making very large amounts. And so that's where we're going to come to when we talk about pandemic preparedness. But it's really a big deal that we'll be able to immediately respond to the next viral pathogen or bacterial pathogen that we're faced with. Turning to therapeutics, we've not been very good at that. There really is just one small molecule therapeutic, and that's remdesivir. Remdesivir prevents viral replication, and it's not a great drug because the virus encodes its own protective mechanism that protects itself against remdesivir. We know that steroids will calm down an overactive immune response, so that's used a lot. And also we have in our hands therapeutic antibodies. In other words, if you're not making your own antibodies, you can actually inject antibodies that were monoclonal antibodies that were synthesized and can be used. Now, what about variants? We've been hearing about them for the past few months, and they're scary. And we need global surveillance. We need all this sequencing of viruses to find out what's out there. What is a variant? A variant is really a mistake that a virus makes when it is busy copying itself inside your cells and makes a mutation. So it changes the sequence of instructions in the DNA and RNA that is then involved. And so the more a virus keeps infecting people, the longer it takes to bring down the viral load in every country, the more mutations will accumulate and build up, because the more you have more viruses replicating. And as you replicate, you get mistakes. You get mistakes, you have variants. And all variants currently in circulation, which are now alpha, beta, delta. Delta is the Indian one, alpha is the UK one. All variants currently in circulation have a higher transmission rate, between 40 and 60% higher. And some, in fact, are more lethal. We suspect that of the Delta strain. It's the most problematic. It's out of India. It is susceptible to the antibodies that you create by a natural infection or by the Pfizer, Moderna, or AstraZeneca type vaccines. So you do get protection. We don't know how long the protection lasts. The people in these companies are already designing the vaccines that will deal with variants by making pieces of RNA or DNA that reflect the new sequence of a variant. So it's something that we have to watch very closely. And the more people that we get vaccinated, the lower the level of free virus floating around, the lower the level of replication of those viruses, and the fewer variants. Vaccination up, variants down. And this is really an important concept for people to understand. Now, I would like to turn to a question that seems to be on everybody's mind that I talk to these days. And that is, do we know the source of the SARS-CoV-2 virus? So let me just do some nomenclature. COVID-19 is the name of the infectious disease. The name of the virus is SARS-CoV-2. Where did this virus come from? And do we know? And are we ever going to know? Well, our alternatives are that it's natural spillover from animals to humans, and not that, but leakage from a laboratory. So let's talk about both. If we talk about natural spillover from animals, there's huge historical precedent. SARS-CoV-1, which was the SARS in 2002, that caused 8,000 cases globally and about 800 deaths. That's a pretty high death rate. That's 10%. Our COVID-19 death rate is down between 2 and 3%. But in both SARS-CoV-1, the natural etiology was bats to civet cats to humans. Another coronavirus, MERS, Middle East Respiratory Syndrome, another coronavirus, was camels. Ebola is bats. So, you know, this is precedent. We see this, and people think that it may be logical, since the coronaviruses are found in many, many bats, that this is the etiology of the COVID vaccine, and that we have undiscovered natural diversity. Between 2012 and 2015, getting samples of viruses from bat caves in southern China, there were 630 novel coronaviruses found in those bat caves, which, by the way, are 600 miles or so away from the Wuhan center of this pandemic. Additionally, we know that there is wildlife trade in wet markets in Wuhan, and there are a dozen wild animal food markets there in a city of 11 million people with an international airport. So it's a hub. People are moving in and out of that constantly. And then people say, well, this thing started in Wuhan. What do we have in Wuhan? We have the Wuhan Institute of Virology, the institute, in fact, that carried out the very heroic and deep analysis of all the various coronaviruses carried by bats coming out of those waves in Yunnan. So, you know, people say, well, wait a minute. Is it possible that some of those 630 novel coronaviruses that were found in the bat caves and brought back to the Wuhan Institute of Virology, in fact, caused the problem? The lab there serves as a crossroads for people and viruses to interact. Is it possible that a researcher or a worker working in one of those labs was infected and then left the lab and infected other people? It's extremely important to realize that the group of people who were first infected, a goodly number had attachments or were related to people in the Wuhan market. It's also important to realize that many others were not, so that we can't make a direct trace from one to the other. In November 2019, which is before the thing really exploded in January, there were 3 institute workers that became ill and were hospitalized with symptoms consistent with COVID-19. We do not have those blood tests. Back when the researchers in the Wuhan laboratories were collecting bat specimens in southern China, 6 miners came down with pneumonia-like symptoms, but they had blood samples, and the claim is that none of them had antibodies to what we now know as SARS-CoV-2. Now, one thing that always surprises me, and I live in this world, is the rate at which accidents happen. Between 2006 and 2013, in the United States, there were 1,500 incidents with select agent pathogens, 800 resulting in hospitalization. By incidents, I mean the safety level protocols weren't maintained, somebody got a finger stuck, somebody's air hose blew out when it shouldn't have. And we've got safety labs that it's called BSL 2, 3, and 4. Biosafety Lab 2, 3, and 4. Biosafety Lab 2 is more or less what I do in my laboratory. I wear a lab coat and I'm careful. When you go up to Biosafety Lab 3 and 4, it's much more vigorous. And by the time you get to 4, you're wearing a full containment suit that you see in the movies. But importantly, what happens at that higher levels is that workers have to give blood. So we always have a way of tracing, if they got sick, which antibodies that were being made. So that's accidents. And accidents happen all over the world, and they happen regularly. So someone will say, oh, my BSL-4 facility, nothing can ever get out of that. It's simply not true. And what we have to worry about is the escape of a virus that we don't want out. That's a pandemic strain. And most worrisome is the escape of an engineered virus. Well, what do I mean by that? Let me talk about something called gain of function mutants. So again, I'm being a biology teacher. A gain of function mutation means that you change, let's say, the virus in such a way that it's going to make that virus more transmissible or more virulent as a pathogen. That's a gain of function. There are many gain of functions that are good, going after something like if you need a vaccine for yellow fever, gain of function mutants gave us that, or vaccines for melanomas. In fact, gain of function mutants help us with that. But basically, there are 2 ways of engineering a gain of function mutation. Some are good, some are bad. But the 2 ways are, 1, genetic engineering, actually chemically changing a sequence to make the virus more virulent, or letting the virus evolve in the laboratory, training it to be more effective at binding to a receptor on a lung cell. So now I'm going to get a little more specific because it's important that we understand this. One of the quote unquote smoking guns for a purposely engineered virus has to do with the structure of that spike protein on the surface of the SARS-CoV-2 virus. When that virus with its spike sticking out attaches to the ACE2 receptor on a human cell, what has to happen is that spike protein has to be cleaved, and then what's left can merge their membranes and get into the host cell. Some coronaviruses have something called a furan cleavage site. Furan is the name of an enzyme that's in our host cell that clips that virus. Now, this furan cleavage site in the spike protein of COVID-19, it was not found on SARS-1, was not found on MERS, but there is one in SARS-CoV-2. And so they say, well, how did it get there? It's possibly it was made there. And in fact, we know that the Delta variant from India is highly transmissible because it has mutations in the furan cleavage site that make it more efficient in infecting human cells. But it turns out that many of the new coronavirus that are being looked at do come naturally with furan cleavage sites. And so it's equally possible that it got into and became part of the COVID-19 guy because it had natural recombination in the wild. So it needn't have been genetic engineering or evolution in the lab. The second smoking gun, and the one that got lots of press, is that when you make a messenger RNA, what it's doing is using a specific group of bases, CGG, to code for a specific amino acid so that you will get a specific protein. And in this furan cleavage site, there are 3 amino acids, 4, proline, arginine, alanine, and the triplet code that is going to tell you to put in an arginine and then another arginine in CoV-2 is the string of bases called CGG. Turns out that this is a rare codon. And so people look at that and they say, oh, wow, rare codon, genetic engineering, it was made in the lab. But in fact, 10% of the arginines in the COVID viruses are coded by CGG. And they are also found in many other coronaviruses. The bottom line is that we have to realize that it's not going to be black and white telling us that the COVID virus either came from natural resources or was engineered or made it into a lab and escaped. One other thing about that furan cleavage site, which really gets me, is that if it was done by bioengineers, they did a piss poor job because it's a rotten cleavage site. And the chances of somebody sophisticated enough to make the damn thing is not necessarily going to make one that's as miserable as it is. So what are our lessons learned for protection from future pandemics? I'm trying to get through this so I can answer questions. So the lessons we learned from what I just told you is that we should stop or control wet animal markets anywhere. We should provide global oversight of gain of function research, and we should provide oversight of the containment facilities at containment levels BSL 2, 3, and 4. This is all no-brainer. The big problem is, how do you police something like this, if not in your own country, which is hard enough, but elsewhere. So I would like to close by just going through the list of things that I think are critical for pandemic preparedness in the future. There will be more pandemics. There is no question about it. It's a question of when, not if, and what should we be doing? What have we learned during this horrendous period of time? And that includes, one, new technology has enabled surveillance. We can sequence the genetic material rapidly and accurately so that we can track pathogens worldwide and identify them early. Make vaccines early, warn people early. Number two, we have to have the rapid establishment of ongoing testing for the spread of the new pathogen. At the beginning of our 2020 pandemic, we did a miserable job. At least we did so in the United States. Other countries did much better. New technology has enabled rapid vaccine production, as I told you about earlier. We need prototype vaccines within days, and we have to understand how to manufacture and scale up rapidly. And that brings me to the next point, is the establishment of a reliable supply chain for making what we need for testing, for vaccination, and for treatments. We need to be prepared in our supply chain for personal safety, ICU equipment, therapeutics, and vaccine dispersal. We also need a plan in place for consistent and transparent communication with the public. Australia did a much better job than the United States on this. We got consistently conflicting information, and that took a tremendous toll on our country. We need to grow and empower national and international groups that speak with one voice. We also have to balance the protection of the economy while protecting the nation's health. And these are not either or mandates. They can be done at the same time. And above all, we need diplomacy. We need to establish lines of international communication about new pathogens, global surveillance, access to data in the cloud, and vaccine distribution. And no country can do this alone. And I want to end by a simple comment, and that is the bugs do not know about borders. They're everywhere. So thank you for listening to this very detailed and hopefully not too complicated discussion about the pandemic. And I'd be happy to answer questions.
Thank you very much, Lucy. That's absolutely wonderful. It seems to me you've covered everything, but I know that there are going to be a lot of questions, and we already have quite a few. So if I may, I'd like to start off with asking a few of those questions on behalf of the people who put them forward to us. One of them, I guess, is there a problem with the quality of the security in research labs, even at the level 4 biosafety level? Is there a need for a different protocol there to avoid spills in the future?
No, I think there's not a need for a different protocol. There's a need for having the protocols enforced. People get sloppy. The BSL-4 safety level should be quite good. And there should be very, very few leaks from that. But the operative word is very few, not none. And while dealing with a pandemic strain, you must always weigh how much risk you're taking against what the good will be by keeping it going. One of the arguments that we hear constantly about, why should we be studying these viruses if they're so deadly? Why should we have them even in our BSL-4? Because we have to understand what a pandemic strain looks like. We have to know how to sequence it so that we can make a vaccine, so that we can design therapeutics. And this is true for BSL-4 and BSL-3. But we have to balance risk. And what is the greater risk? What is the greater benefit? The risk-benefit conundrum is very strong here. Mistakes happen. Things get out. I know of a case in a BSL-4 facility where one of the hoses that was connected to the oxygen came loose. Somebody died because of that. And, you know, these things happen. You know, a colleague of mine, this happened several years ago, died from an infection that he got in a laboratory with something he was working with at a BSL-3 facility. Accidents happen. And we don't have global or even national control of surveillance of these biosafety labs. But people are trying to be careful, but remember, we're human. Mistakes happen. Yeah. We're not machines.
This it comes back to what you reinforced at the end of your talk, which was the need for a much better exchange of information across the world and international collaboration. And one of the other questions that's come here from Samuel, he really addresses that. He feels that only division and debate seem to be coming out of what we are learning from the pandemic. That's a rather pessimistic view. He doesn't think the WHO, for example, has very much authority. And so the question, I guess, to you is, do you see a need for a new kind of approach to the international exchange of ideas and information?
Yes, I do. I agree with him. And I think we're learning. This pandemic showed us that we're really in pretty miserable shape in being prepared for pandemics, and we better change that. And also, we should really take advantage of cloud computing and get all the information we possibly can into the cloud and accessible to everyone in every single country. Now, we turn to the etiology of our current miserable virus, and we say, well, are we ever going to know? Is China ever going to release all the data and blood samples? Is it really all a problem in China? No. Many people are involved in this all over the world. And we need some forum that is open to discussion and understanding. Accusation right now is useless. We need facts. And that's what we have to aim for.
Thank you, Lucy. There's another question here from Suzanne Corey, whom you know from the...
Hi, Suzanne.
She'll be delighted to hear that. Suzanne says, thank you for your fascinating overview. Please comment on how we can improve the scale of vaccination development and the future of vaccines perhaps being delivered as a pill.
Yeah, so I think that scaling up vaccine production is absolutely critical, because we're going to get more and more variants if we don't. One of the object lessons came out of the Serum Institute in India, where they had scaled up the AstraZeneca vaccine way before it was even through phase 3 trials. And they had sold a lot of their vaccine to other countries. And then they had a horrendous spike of infections in India and didn't have enough for themselves. So when you say, how do we go about scaling this up? I think this has to be done as a global community. I think that we have to understand that vaccinating everybody in the United States is not going to get us to herd immunity or anywhere near it. And until we have a huge percentage of people globally vaccinated, we're not safe. And I don't think this message has come through to them clearly enough. And the race to make more and more vaccines is on. I also have to credit Cuba for the incredible work they did on their own, making their own protein-based vaccine and not relying on anybody else. And that turns out to be 92% effective. So I can't answer you in any very, I don't have a menu for how to do this, except that we've got to do this. With respect to a pill, I know that there are people working on it, and it's very far from a reality. Unless you know something.
I certainly don't. Let me come to another question, which is from Susan Pond. Susan, for your information, Lucy is also a scientist. She's a very distinguished scientist who's had a brilliant career in Australia and also overseas. And she's currently president of the Royal Society of New South Wales. Susan says, does Dr. Shapiro categorize the adenovirus vaccines as GMOs? I guess she means genetically modified organisms. What are the additional regulatory oversight, if any, needed for these vaccines compared to the mRNA type vaccines?
Well, there isn't a lot of difference between the mRNA vaccines and the adeno vaccines. The main difference being that one is packaged inside a harmless virus. The other being that the mRNA is packaged inside a little lipid sac. It's just the delivery. Okay. So, in both instances, you have genetically built a sequence that is coding for a spike protein. You're just doing it in two different ways. And yes, there's a great deal of oversight that has gone on in both instances.
Thank you, Lucy. Can we also talk a little bit about supply chains, which you referred to earlier? A question here from Anwara. How will strategic decisions minimize supply chain uncertainty with future pandemics?
Well, I think first of all, we have to identify the weak spots. Take a given country that had stopped making pipette tips that you need in the laboratory. And you're dependent on, I'm making this up, you're dependent on Thailand for the pipette tips. You're dependent on China for something else. You're dependent on Russia for a third thing. You're dependent on Australia for something else. We have become so completely dependent on our supply material on other countries because we have never been faced with the just-in-time dilemma. People are not stockpiling anything. And so now we have to learn that there are some things that you must stockpile. So in a sense, this pandemic has been a learning experience. And hopefully we'll act on it. The problem is acting on it means changing your economy, getting your hands on money, making things that you haven't made in years and years and years, changing very fundamentally the economic base of a given country. So this is not minor. And I agree with the person asking the question. It's a huge issue.
We had some discussion of this question when we were interviewing Michael Spence about the impact of a lot of the things that have been happening recently on globalization. And this is certainly part of what you're saying there. Yes. That's perhaps a sequence into another question from an anonymous attendee who says, thank you, such a wonderfully structured and delivered presentation, which it clearly is. It seems as if we could be years before the world gets back to normal. And I'd be interested in your view of what the future normal will look like. Will we ever get ahead of these viruses?
I think we will, but it'll be a new normal, which is what we all talk about. I think that with changing supply chains, changing economic base, appreciating surveillance, global surveillance, appreciating the fact that these viruses are going to be coming with increased frequency is going to skew how our global health programs going forward are carried out and place them on a much higher international scale than they are at the moment. No man is an island, no country is an island, not when you're dealing with pandemics. And so that will change. The other thing that I'm finding changing here in the States is the way in which we work. So here we are on Zoom. I'm talking into everybody's living room and bedroom and bathroom at this point, one-on-one. This is new, but we're learning that it works. I am able to talk to you. I didn't have to get on a plane, although I would have loved to visit Sydney, where I've never been.
Next time.
That would have been wonderful. But we are able to communicate. And I know that in the Beckman Center at Stanford now, I am the director of the Beckman Center, and we have just put in place a new way of coming to work. People are going to work remotely 3 days a week, and come in 2 days a week. And it can be variable depending on meetings and what you have to do. But we're now moving to a very different kind of work plan. And remote working and the technology of communicating like this is only going to get better and better. So I think there's going to be a new normal. Anonymous questioner.
Good question, and a very interesting answer. I know, Lucy, you mentioned previously in a conversation that you had actually published a large number of papers during the last year, despite the pandemic, with colleagues.
Mm-hmm. You froze for a moment, so repeat that.
You had mentioned in a previous conversation that you had published several papers during the last year with your colleagues.
Oh yeah. And look, I have, my lab has gone on. The way in which the people in the lab have worked have been on shift hours. So there's never more than 1 or 2 people in a lab at the same time. It runs 24 hours. Wearing masks 6 feet apart. But students and postdocs are in the lab. They are working. The professors who are older and more susceptible are sitting on their computers. But I found that I was able to do really a pretty effective job of communicating with everybody constantly, because I don't have to walk across campus or fly anywhere. I'm here. And I've also, it's been amazing to me that I sit on several corporate boards, and those boards have met by Zoom. And by golly, it's worked. We got a tremendous amount of work done. We've done some really good stuff. So I must admit I'm somewhat surprised. Do I miss the personal interaction? Sure I do. I miss having a cup of coffee with someone or a cocktail in the evening. And I think slowly those things will come back. But it's going to be a while before we're most comfortable in doing that. And it all depends on vaccination. And then while it depends on vaccination, it also depends on how long our immune response lasts to these vaccines. We don't know that yet. So there's a lot of surveillance that has to go on.
A lot of unknowns still, despite.
We have a lot. Yeah, I think that's been the most worrisome thing, is learning to live with unknowns, not having real answers, trying to weigh different possibilities for everything. The safety of our children going to school, the safety of our economy, where we're going to be, will our postdocs get a job? What is everybody's future like? Yeah. And it's scary, but I think we're all working on this together.
Just before we end, Lucy, I think a number of people on this call would be very interested to know a little bit about your business efforts, how you took chemistry of boron into new products. Could you say a little bit about that, please?
Sure. I'll try to be brief. I must be about, almost 20 years ago now. It occurred to me that the world was in terrible shape and that we had emerging infectious diseases, increased antibiotic resistance. The major pharmaceutical houses were not making new antivirals and new antibiotics. And it just didn't look good. And so somehow, those of us who were trained in this business should help. And so I got a hold of a brilliant chemist named Steve Benkovic from Penn State. And we wrote a very short proposal to the Department of Defense, DARPA, and said, we're going to design new antibiotics and we're going to tackle antibiotic resistance. And what we did was we looked at the periodic table and we said, oh, look, boron is right below carbon. What if we made a whole series of small molecules that were boron based? And Steve Benkovic went ahead and built all these little molecules under 300 molecular weight. In my lab, I had all these pathogens lined up, and yeast cells and HeLa cells, human cells. And I found that our boron compounds had incredible activity in inhibiting the growth of many pathogens, but not touching the human cells. And so we did the critical experiment, and Steve replaced the boron with carbon, and we lost all activity. So the first thing we did was hire a lawyer and patent everything. Second thing we did, because it coincided with an anthrax scare in 2001, we decided to apply to the government for aid to start a company, and we did. And that led to Anacor, and Anacor brought us to the first antifungal with an entirely new mechanism in 50 years that received FDA approval. And then we went on and made other drugs and ultimately sold that company to Pfizer. The money we made from selling that to Pfizer, we decided to use our boron library to help in the world of agriculture, because fungal infection of our food crops, like bananas and soybeans and wheat, is a huge problem in the time of climate change. So we started a new company called Origin, and we've been working on that. And just recently, we have spun out of that 3 separate companies: a crop protection company, an animal health company, and now going back into human health in infectious diseases and RNA viruses. That's succinct.
That's very succinct and fascinating. And perhaps we'll have to have you talk again, Lucy, about business ventures, because there's a lot you could teach us on that as well. One final thing before we stop, because of time. I know that in your early career, you didn't start your university work in the field of biology. You were actually in a completely different area, literature and art.
Yes.
Can you say a little bit about what drove you to do what you currently do?
Well, I think there's a lesson there. And yes, I was a fine arts major, and my senior thesis was on Dante and why he wrote in the vernacular and not Latin. And the lesson is, I'm watching my grandchildren who are now entering college, worrying about, what am I going to be when I grow up? What should my passion be? And I'm going to college right now, and I should know now. Well, not necessarily. Receiving a very broad education is a wonderful gift. And if you can manage to do that, do that. I didn't really understand my passion for chemistry until quite late. But when that hit, when I finally took organic chemistry, it was the most beautiful thing I'd ever seen. And that changed my whole life. But that happened late. It happened when I was graduating from college. So it's a lesson to young people. You don't have to know from day one of university who you're going to be when you grow up, or even what your passion is going to be.
But be open and interested and curious about other fields.
Of everything, yes.
Finally, you've spoken quite a bit about the information content of biological organisms. And I think your own original work was looking at a single cell organism and understanding how the cell develops over time. And could you say a little bit about how information and the concept of information in the cell came to you, and how you've developed that interest with, I understand, with your husband, Harley?
Yeah. So Harley came in midway, sort of in my career. When I first started out, I had one overriding question, and that is, how does a cell function as an integrated unit? A cell has to do a lot of jobs, and it can't just be randomly doing things. It has to be some sort of integrated system. copying the DNA, dividing the cell, carrying out metabolism, growing things, suborganelles on the cell. It had to be integrated. And the other thing, to me, that was an outstanding and important unanswered question is how the cell uses positional information, where to put things in a three-dimensional cell to control this integrated circuit that runs it. And so I was busy through the years building up all the understanding of the biochemistry. And then Harley came along, and Harley is a physicist, Bell Labs, and he said, my goodness, this is just like an electrical circuit. And so then we were able to put all these things together and use electrical circuitry as a paradigm for looking at a cell as an integrated system that includes both temporal and spatial control mechanisms. And we were able to carry this out in its fullness for a simple bacterial cell.
Wonderful. Thank you very much, Lucy. I think we're running out of time, so I'm going to have to end this wonderful session. On behalf of everybody who's been listening, I think we can say that this is the first time, and certainly speaking for myself, that I've had such a clear and comprehensive understanding of what COVID has been, how it originated, how it's progressing, how we're dealing with it, and why these new vaccines are working, and what is the mechanism. So thank you for doing that. I think we're all enormously grateful to you for what you've done today. It's just a wonderful presentation. I hope we'll have a chance to see you again sometime before too long. And on behalf of everybody here, thank you so much for what you've done. Thank you.
It's been a pleasure, and thank you for listening to me. Take care. Good night.
Thank you to all our participants who've been listening today. Thank you so much. And please join us again for the next sessions we'll be having very shortly.
Bye-bye.
Bye, Lucy. So, ladies and gentlemen, there'll be a little bit of information here about future events that are coming up. And also, I should mention that those of you who may have missed some of our past events with Stanford professors can certainly find those on the school's website. So we look forward to seeing you again in the future, and we hope that you'll be able to participate again. All the best. Bye-bye.